The RAS gene mutations are very common in cancer. They occur in about 30 to 40% of all cancers. And in fact, it is the first oncogene discovered and nearly about like 3 to 4 decades ago. So KRAS is the most common isoform of these mutations and has been the target for drug development for several decades now. One of the reasons why it is so hard to target KRAS is because mutant KRAS protein binds to GDP and GTP with high affinity. And they lack the surface hydrophobic pockets that make it really hard to develop nucleotide competitive inhibitors. So the recent advances in medicinal chemistry led to the development of some covalent inhibitors to target this reactive cysteine 12 residue of KRAS G12C mutant protein. And this could disrupt KRAS-driven proliferation in the cancer cells. So this CodeBreak 100 trial represents a clinical validation of these efforts to target KRAS G12C. And this is a major disruption in the management of patients with KRAS mutant lung adenocarcinomas. So this is a phase 2 study, the CodeBreak 100 clinical study, and this is evaluating Sotorasib in 126 patients across the world in 11 different countries. And there were a total of 120 patients who had centrally valuable lesions. And in the study, the overall objective response rate was 37.1%. With three patients having a complete response and 43 patients achieving a confirmed partial response. And these data are similar to the data that was presented early on in the phase 1 part of the study. And the safety was very identical to the phase 1 study with Sotorasib. The findings are a great validation of the fact that we can actually clinically target KRAS G12C with an inhibitor that is very selective and with an excellent safety profile. And of course, there's a lot of work to be done. There are a lot of different collateral mechanisms for resistance to KRAS G12C. There's a lot of effort going on right now to understand the resistance to these inhibitors, and there are a lot of potential combination-based approaches that are being currently studied.